Good morning, students. Today we'll start discussing on protein secondary structure prediction. So this is actually the part 2 of our specific lecture on protein secondary structure prediction, and here we will mostly see the exactly the state-of-the-art or benchmark algorithms that are in use, and some of the methods, some of the features, how we actually implement protein secondary structure prediction in our regular applications, how benchmark data sets are validated, etc. So I'm now going to present the screen. So protein secondary structure prediction, this is the second part of our lecture series. As I have just now mentioned that today we will see standard methods and some benchmark algorithms, more specifically on starting with PSSM, position specific scoring matrix, one of the most popularly used feature and profiling method of the sequences that we need to predict, that we need to train. So it is just a revisiting of our earlier discussion on PSSM, and we'll see how PSSM and physicochemical properties are clubbed together and used with standard machine learning algorithms to predict the secondary structures in proteins. And there are many different methods which are popularly used, and we can see that PSIPRED is one of the most popular ones, then SIMPA96, PhD, Predator, and NNSSP, HydroSP, JPRED3. Then there is a method called PSP-MCSVM. This is one of our developed methods, developed by Chatterjee and others. So this is also giving very interesting results. So this is also a widely cited paper in public domain, available in public domain. So we'll also discuss this. Along with that, we'll also develop, we will also discuss a series of GOR methods, GOR 1 to up to GOR 5. So these are also very popularly used benchmark methods. And finally, a brief overview of PorterPaleAle. This is one of the best servers available today for protein secondary structure prediction, for giving in excess of 80% prediction accuracy. So starting with position specific scoring matrix, as you have already seen that when we have sequence information, we can actually calculate its position specific frequencies and position specific probabilities, like as definition says that PSSMs are constructed by calculating position specific scores for each position in a multiple alignments of with the highest scoring hits in an initial blast search. So if we have a specific target sequence, and if we just put it in a blast search, that means if we do multiple sequence alignment, and if we have some 10 sequences found that correspond to that target sequence, and we can actually identify that for a particular position, what are the other possible, what is the probability of a particular amino acid coming to that particular position. So this is kind of frequency based scoring scheme. So we can actually generate a position specific scores, which is called PSS, or there are different names, like for example PSSM is also called as PWM, position weight matrix. So position specific scores are calculated by assigning high scores to highly conserved positions and near zero scores to weakly conserved positions. For example, a particular amino acid may be having a high propensity to appear at a particular position and gaining a particular high score. So this is a very interesting feature. We'll see one example quickly. And The profile thus generated is then used to perform a second blast search, and the results of each iteration is used to refine the profile. So we can actually use this blast as an iterative process to refine the overall result, and we can eventually get a very refined PSSM matrix. Now, protein sequences are transformed into FASTA format, and using say PSI BLAST or some other algorithms. So in general, when we talk about PSSM, it is a very simple concept. We can actually take up a very standard even nucleotide 4-nucleotide example to make it simple, and we can see that how the frequency scores can actually reflect into a position specific scores. Okay, so let us see one example that say we have a target sequence, and we give the target sequence to BLAST, and we generate a set of, say, you can see that 10 different sequences are generated by the BLAST server, say, for a specific iteration. And then what we need to know that what is the how many times G is appearing at the first position, how many times T is appearing in the first position, how many times C is appearing in the first position, and how many times A is appearing in the first position in the whole search. So we can see that the corresponding position frequency matrix of PFM can be constructed as, okay, A has appeared 3 times, G has appeared 1 time, and T is appeared 4 times. So this is the general frequency, and you can see that here, for example, at this position, G is appearing 10 times. So that means this particular position is highly conserved, and this here T is appearing 10 times. So you can see that this G and T are highly conserved across this, and based on the values, we can actually have a very interesting insight on which regions are getting conserved and which are not. So as you can see that in some cases, like for example in the first case or maybe in this case, you have very high volatility and not even a single value is having more than 50% propensity. So you can see that this is a less conserved region, and this is not conserved. So what you can do, that you have 10 values or 10 sequences aligned here, and you have the position frequency matrix, and you can construct the corresponding position probability matrix or position specific scoring matrix, PSSM, as like like 0.3 by 10. Like here you have 10 sequences, so it is like 3 by 10. Here it is 2 by 10, and in the simplistic case, you can see that sum of all this should be 1. Okay, so if you have m number of sequences, like the sequence length is m, you will always get m cross 4 ppm or PSSM. So if you talk about secondary structure prediction problem, so you have 20 different variations. In case of ACTG, you have 20 different amino acids. So whatever may be the length of the sequence, you will always get m cross 20 matrix. So that is the advantage that you submit a target sequence in a multiple sequence alignment server like BLAST, and then you get a corresponding position specific scoring matrix. So that is very frequently used as a feature, and we'll see how we can utilize this efficiently for our prediction problems. Along with these position specific scoring matrix, we also have physicochemical properties. Physicochemical properties are utilized from like there are several databases. For example, you have some standard chemical properties like molecular size, maybe polarity, or maybe hydrophobicity value. And also like the propensity values or relative frequency values of amino acids in helix and shape, which are very important properties for predicting underlying protein structure. So there are a lot of databases available. So you can have a index database release 9 or whatever may be the current release, like maybe I have there is a very recent release of 10 as well. So here you have close to 550 odd properties, physiochemical properties listed against every amino acid. So you can have, for example, a hydrophobicity value for each amino acid mentioned there, or molecular size of each amino acid mentioned there, polarity of each amino acid mentioned, and not just single hydrophobicity value. There can be different hydrophobicity values estimated through different types of computational or biological, not biological, chemical processes. So you may need to use say 3-4 different hydrophobicity values as your features. So different types of physicochemical properties are utilized in association with statistical features like PSSM for popularly, predicting the protein secondary structures. In association with that, there are other structural informations are also used, widely used, different types of structural informations. For example, like informations from 3D structures, like your PDB, like protein data bank structures. So all these informations are also widely used for protein secondary structures. But I am just giving these two examples, and let us directly go into different methods and let's see how they are actually utilizing these properties for prediction problem. They are popularly used in conjunction with different different machine learning algorithms like neural networks, support vector machines, random forest, or maybe hidden Markov model. So coming to PSIPRED, PSIPRED is a very popular protein secondary structure prediction method widely used even today, and it is a neural network based method which has basically 3 components, and what it does, it heavily depends on the homology modeling, because it needs to, like, whenever we want to use profile features like PSSM, like it is more like a statistical profiling. So we usually need to do some kind of homology assessment, like when we need to identify the secondary structure of a protein, we need to know what are the available or similar type of sequences available in the literature. So we first do a PSI blast that gives you a alignment score, or you find a set of homologous proteins which are having strong conserved regions. So if you have strong conservations, then you need to have, you become pretty sure that in those conserved regions probably the structures will also be conserved. So that means the secondary structure predictions become easier or more predictable. So we start with PSI blast and get the set of homologous proteins. Then there is a process which is called sequence to structure prediction. Okay, so this is sequence to structure prediction is a method which uses a standard backpropagation neural network, where in input you have the 15 residues are used as a window. If you remember, we have always discussed like a sliding window technique. What do you mean by a sliding window technique? That means that you cannot just predict about a single residue by its own, because as you have seen in case of alpha helix, there is always a 6 to 10 amino acids, they form a specific structure together, because there are hydrogen bonds that form in in the spiral, or similarly in case of beta sheets, there are hydrogen bonds that form between neighboring or subsequent amino acids. So it always is helpful to consider a window of amino acids and extract the features for all of them, but we predict the secondary structure for the central one. For example, if we take a window of 15 residues, that means we are predicting the secondary structure of the central residue, and we have 7 residues on the left and 7 residues on the right. So it is a 7-1-7 structure. So for prediction of the central residue, we take all the 15 residues in consideration and extract features from all of them, and eventually we try to predict for the central one. The neural network has architecture is like 75 hidden nodes and 3 output nodes. Okay, and the output of the sequence to structure network is fed to a structure to structure network, again with a window of 15 residues. That means it's a 2-stage cascaded network, where first we are predicting from a sequence information to the secondary structures, and then again from the secondary structures, prediction probabilities of the secondary structures are considered again in the 15 residue, and again they are predicted with a structure to structure network. So input is also a structure with the neighboring information, and again we are predicting the structural information. So this is the basic PSIPRED architecture, which achieves somewhere around in excess of 75%, in between 75 to 80% prediction accuracy on the independent test set. So as we see that the prediction method is split into 3 stages. So first we generate a sequence, then the PSSM profile, as we have seen that the PSSM profile is generated, and prediction of the initial secondary structure is generated from the sequence to structure network, and then the sequence to structure network result is filtered using the structure to structure network. So this is basically a neural network architecture and standard feed-forward backpropagation network architecture with a single hidden layer is used for PSIPRED. This is the general architecture that we and a superficial idea of PSIPRED that we are providing here. So as you can see that we are using a 15 amino acid window, and there may be a question, why 15? Why not 17? Why not 13? Why not 9? Why not 21? So this is a heuristic approach. So people actually experiment with different windows, and you can actually see that in which window you get the optimal result. So in PSIPRED, they have shown that okay, with 15 amino acids, they could achieve the best results, and these set the benchmarks. Okay, so a lot of people can refer that okay, PSIPRED they have used 15 amino acid window. So okay, you can also apply on a 15 amino acid window. You can also vary. We will show that different approaches use different types of window length. Sometimes it is because of the features that they are actually using. It is not always on the it's not always on the specific you cannot say that this is always absolutely true for across all different methods. Maybe this is dependent on the features that you are using, the physicochemical features that you are applying here. Okay, so here what will be the input number of nodes in the network? So we have used PSSM profile, and we have 15 window. So basically we have actually 300 features coming from 20 cross 15 we have 20 features coming from the PSSM profile, and 15 amino acids and 20 features. So that means it is a 15 plus 20, that is 300 inputs coming from there. And there is one extra unit that is coming from, like, whether it is spans either from N terminus or C terminus. As you know that in the protein backbone you have N terminus and C terminus, and that whether it is close to the N terminus or C terminus, based on that you add one extra feature to that. So for every amino acid you are actually taking 21 features, and there are 15 amino acids to use, so you are actually using 350. So if you ask me that, can you please explain it again, that from where we are getting 20 units for the PSSM profile? So I can go back to the PSSM chart, and I can show you that here, for example, if this is a DNA sequence example, because of short example. So if this is if these are amino acids, so you'll have, here we have 4 rows, there you'll have 20 different rows. So for G, if this is the target sequence for G, you get here 4 different features: 0.3, 0.2, 0.1, 0.4. So if you have 10 different amino acids here, so you have 1, 2, 3, 4, 5, 6, 7, 10 different features you get. 10 different amino acids you will get here. And for every amino acid, how many features you are getting? Here we are showing 4, but in case of 20 features, 20 different amino acids, you will get 20 for each. So if you have 15, so you have 15, for each of the 15 residues, you will get 20 values in the column. So you get 20 units for PSSM profile for each, and you get 1 for this NC terminus. So ultimately you get 315 features estimated for the whole window of 15 amino acid, and all these are done to predict the secondary structure of the central amino acid, that is the 8th in the sequence in the total window. So this is the general idea, and as you know, how the standard neural network work with, like, multi-layer perceptron with backpropagation algorithm. They have used 75 hidden layers in the single hidden units, the single hidden layer. 75 units were used, and in the output, what you predict? So you predict either helix, strand, or coil. That means helix, beta sheet, or coil. Coil means anything other than helix or strand. So basically you have only 3 units as in the output layer. And there are standard training procedure. They have used 0.9 momentum factor, learning rate of 0.005 is slow learning. You can also play with many things, and you can incrementally change the learning rates, etc. To prevent the overfitting or overtraining of the network, what they did, they always use 10% of the training set on which they can optimize the parameters and to check, like, where to stop. So 10% training set was kept to evaluate the performance of the network. Okay, and the training of the network was halted when you get the optimal performance on the 10% that excluded, that is called the holdout set, and then you finally evaluate the performance on the test set. Okay. And to produce the training and test sets, so how they produce it is also very important, like which proteins you consider for the training set, which proteins you consider for the test set, so that there is no overlap and there is no such proteins that have even some kind of homologous. They may be different, but they may have similar homologous properties between training and test set. To take care of that, what they did, they used the Cath classification for the constituent domains. Any protein in the training set which had a domain or fold in common with that of the domains found in the test set are excluded. So as we discussed before, that the domains or the folds are the main active units in a protein. So, and the domains are significantly conserved. So even if the proteins differ, if they have a common fold, they may have significantly same properties, and the whole training may be a biased one. So they have excluded all the common domains from the training and test set. And the further check was performed using 5 iterations of PSI BLAST to detect any missed remote relationships that might not be represented by the Cath classification scheme. So along with that Cath consideration, they also look for homologous sequences using PSI BLAST 5 iteration process, so that there is no common protein in the trainings in the test set that is available, that is present in the training set. And the independent training and test set pairs were compiled using highly resolved structures, like so that you have less than resolution of 1.8 angstrom, and they are highly gold standard. Like that, you mean to say, like when you have a predicted structure with such a low resolution or such a high resolution, actually that means the X-ray crystallographic experiment could very accurately identify the true structure of the protein. So we only they only conserved they only considered only 187 proteins in the testing set. Those have very highly predictable or very highly accurate structural information with resolution, with all the experiments conducted with less than 1.8 angstrom extracrystallographic resolution. That means you can have the structure identified with less than 1.8 angstrom difference. You could identify up to means even less than 1.8 angstrom difference. So that is very high resolution, and they identified 187 proteins in that and divided into 3 sets, 62 and 63 chains. And as you can see that they could actually get very high Q3 scores. Around 25 proteins were identified with close to 80% Q3 score. So what is a Q3 score? The Q3 value is a fraction of residue predicted correctly in the secondary structure states like helix, strand, and coil. So you have you actually have prediction of helix, strand, and coil. So you can have only for QH, which if you want to know the prediction score of helix, you can have QS, that is the prediction score of strand, or QC, that is the prediction score of coil, or Q3 is the combined prediction score of all three. So it is important, because when we predict a whole protein chain, then we need to know that what is the overall prediction score. To determine the overall prediction score, this is often termed as a Q3 score. So you can see that yes, there are some chains which are predicted at very low accuracy, and again some are predicted at very high accuracy. But in general, you can see that 72, 74, 75, 78, 80, 82, 84. So up to 90, even up to 90, there are 5 chains predicted at a rate of 90% of accuracy. And SOV is the segment overlap score. So this is also very important score, that how many sequences have a significant segment overlap has been. So this is also a very popular method, that segment overlap score has been used to evaluate the predicted protein secondary structure, that is helix, strand and coil, by comparing with the reference secondary structure. That is another setup. Like, you have a reference structure and you have a predicted structure, how much you have the overlap. Okay, so this is the general prediction of overlap score. So Q3 is individual prediction and SOV is the overlap score. So if you consider the SOV score, you can see that there are like 76%, you have 16 sequences with 76% SOV score. Okay, around 76% SOV score. So 80 up to, you have 96% SOV for close to 5-6 test proteins. So this is the result of PSIPRED. That is a very standard and benchmark method that is used even today. And they have also done extensive testing on CASP3s. There are often benchmark testing datasets released, and CASP is a benchmark dataset, and CASP3 dataset, like target proteins, they have Q3 scores for alpha and beta. That means for the helix and the beta sheets, see up to 78, 90, 96, and average by residue, you have 75% residue and 77% for beta sheets. And SOV scores are also like 70 to 80% in general. And you can see the segment lengths are sometimes they're short, like in 70s, and in most cases they are up to say 300 plus sequences are also there. So these are general idea about how these methods perform with the help of say features like PSSM and some standard machine learning classifiers like neural network. Okay, so with this, let us move to some other methods. There are actually many different methods available. So, for example, SIMPA is another very popular protein secondary structure method developed by a group of Rost and Sanders. So they used again window-based method for short segments, and they used multiple sequence alignment with appropriate cutoffs to supply more enriched information about the protein secondary structure. So SIMPA 96 actually used a second level network that is called sequence to structure, and then again structures to structure network. And finally, the interesting part in SIMPA is they employed a voting method. That means they used a consensus strategy. The consensus strategy is designed from 12 different neural networks working in parallel on the same input, and they try to vote for a specific decision. So SIMPA generated reasonably good, close to 70% result in 7-fold cross validation and 3-state prediction accuracy. Okay. Another method is by again by the same group, Rost and Sanders. They were working on a method which is called PhD, and this method takes sequence profiles that means the PSSM, the conservation weights, insertion deletion, and spacer, that means when you have multiple sequence alignments. So what are the insertion deletion frequencies and what are the gaps? So everything they have considered, the length of the protein, distances of the window with respect to the protein ends, that means the C and the N terminus that I was mentioning. So they have included all these as features, and again on a benchmark RS 126 dataset, they achieved accuracy of 73.5%. More methods like Predator, NNSSP were proposed. In Predator, why I am showing this is like you can see that the progression and what are the different types of features people are using. So in Predator, propensity values of 7 secondary structures and local sequence alignment scores are used in lieu of global multiple sequence alignment. So Predator used local sequence alignment rather than global sequence alignment, and they could achieve around 70% accuracy. NNSSP is again called a third generation method and scored nearest neighbor method by considering proteins from the again the N terminus or C terminus in helix and strands, and like when it is a helix, you can actually estimate that what is the distance from the one end, whether this is a N terminus or a C terminus, because you always have a backbone which starts from N terminus, and one side you have N terminus and another side you have C terminus. So if you have a helical structure, so based on its distance from the N terminus or a C terminus, there are differences. These are also considered as interesting properties, and people have encoded these as features, and they have got interesting performances. Like in case of NNSSP, they have achieved close to 73%. HydroSP. HydroSP is another hybrid method which combines the knowledge of there are often meta approaches. Meta approaches or hybrid approaches are approaches which combine several existing approaches. So HydroSP, instead of developing everything from scratch, they try to utilize the properties of the strength of PSIPRED. Okay, and they used a knowledge-based approach, ProSP, along with the machine learning-based approach, which is already available as PSIPRED. So ProSP is a knowledge base considered on small peptide fragments along with the structural information. What is this? Is that you have different conserved regions, and for which we may know the exact secondary structure information. So if you have previous knowledge that this small fragment is having this particular secondary structure information, so you can actually encode that and generate a knowledge base. Okay, that this sequence, if this is the sequence, you have this structure. So there is no prediction involved here. You only collect all the available knowledges from the available PDB. Protein Data Bank is a storehouse of secondary structure, gold standard information. So they are just combining the knowledge information from there. And they are combining that with PSIPRED. So a quantitative measure called match rate is used for a target protein to extract structural information from ProSP. A match rate of at least 80% is then information is being used for prediction. So you have a target sequence, ProSP is a knowledge base, and you have a you make a query to ProSP, and if you get 80% match, then you can actually use it for your prediction. And then you use either ProSP or you use PSIPRED. So you use them as a hybrid tool between ProSP, which is knowledge based, and PSIPRED. Because sometimes you may give a target sequence and there may be a situation that such a sequence information is already available somewhere. Significantly conserved regions are available somewhere. So in that case, just a match with a knowledge-based approach may give you much better result than a prediction-based approach. So that was the concept behind HydroSP. So you can see that the average Q3 score of HydroSP2, the second version, is in excess of 80%. So it is 81% and 80%. So these types of meta approaches are often, or hybrid approaches are often really useful. Now there are some more methods which are called like JPRED3. JPRED3 is powered by a JNet algorithm in which position-specific scoring matrix again and hidden Markov model is used. In other algorithms, mostly we were seeing that either in some cases support vector machine, in some cases neural networks are mostly used. But here hidden Markov model is very popularly used, as we discussed in the last in part 1. Hidden Markov model is widely used in protein secondary structure prediction. So it developed a 7-fold cross validation experiment with scope domain data, and you achieve a Q3 score of in excess of 80%, that is 81.5%. Then comes another method which is called PSP-MCSVM. So this is a method developed by PLE Chatterjee and others, like we are also part of that group. So this method is very interesting, and I am presenting this because this is also having very good citations globally, and people are also using this for this type of two-stage methods for secondary structure prediction approaches. And here a two-stage approach is used. So just like PSIPRED, what they did in the first stage, classifier is designed to predict sequence information to structure information, and then in the second stage, structure information is mapped to again a structure information. So how is this done? So we'll discuss this briefly. That in the first, features are extracted from amino acid residues belonging to a hypothetical sliding window. So what do you mean by that hypothetical sliding window? As I said, in some cases they have used 15, some cases they have used 19, some cases they have used 21. So in different applications, people use different types of windows. So here, a sliding window based technique has been used, and as features like position specific scoring matrix is used, then hydrophobicity value, molecular size, polarity, relative frequency of amino acids in like the propensity of amino acid is used for each amino acid in the sliding window. And MCSVM classifier 1 predicts the secondary structure of the central amino acid of the window. By sliding the window over the entire sequence of amino acids and repeating the classification process, secondary structures, the sequence helix or helix sheet or coil of the entire protein chain are predicted. So this is the first classifier's output. Now, sometimes what happens, like the first classifier's output is often not completely pure. You may need to refine that, because even if it is considering the neighborhood of the sequence, neighborhood sequence information, it is not considering whether suppose you have a helix predicted and either side you have sheets, so that is not proper, no? So even in some cases, you need to filter the secondary structure prediction results. So to refine the structure prediction result, so they have used again a sliding window based on the information from the classifier 1 to predict the refined secondary structure information. So that is called the MCSVM classifier 2. So that gets better refinement over the prediction decisions about the prediction over the secondary structure annotation for the first classifier. So refinement based upon contextual information about the secondary structure estimated over the neighbors of the central residue within any sliding window. Say we are taking say 9 sliding window, then for every amino acid or every residue or every position, there may be 3 types of classification decision, that whether that is a helix, sheet or coil. So you have 3 different strengths or 3 different prediction scores coming from the C1 classifier. So that information is used in C2 classifier as features, and again in the output they are predicting H, E or C, that means helix, sheet or coil. So that is the approach. As you can see that amino acid sequence informations are used, and you have multi-class SVM1, and you have the second set of classification decisions, and you have these taken as features, and then you take what is the maximum confidence value of a particular classifier, particular position as H, E or C, and based on that you give the final output as a secondary structure. So this is a multi-class SVM for protein secondary structure prediction. And as you can see, the results are also very close to 80% in many cases. In the results are ranging, the Q3 results are ranging between 75 to in around 75%. So as you can see, the helix prediction rate is very high, close to 80%, and in this case also you can see the helix prediction rates are very high, and SOV is around 70%. You can see that this is still a bit lower than PSIPRED, but it is still much better than the methods like SIMPA, DSC, and others. So this is also considered as a good approach and widely used by other people to advance, and especially the approach is highly considered, and many people have built up on this method. Then there are other methods like GOR. GOR is a typical method which is an incremental development procedure. So what they did, they used different known structure informations for prediction of helix, sheet, turn and coil. Okay, for each position, and based on a 17 amino acid sequence window. So each of the possible states structural information supplied by the neighboring residues of a central amino acid are summed up. So you have a central residue and you have structural information from your neighbors, and you just sum up all the structural informations and you predict for this method. So it started with around 60% accuracy, but you know, like as the method, the basic methodology of this particular GOR technique, it says that it started in the GOR 1 with the 75 known proteins only. Okay, so they used 75 known proteins structural information to compare the structural information of the unknown structures. Okay, so in the subsequent methods, they kept on enriching or enhancing the data tables and combining more and more structural information into the subsequent methods. All such information combined with the structural information preconditioned with the existence of particular amino acid in the central residue under GOR 3. The GOR 4 difference between information sum of the structural state and the for the set of all complementary states is considered before determining the state of a central amino acid. In GOR 4, for example, they use 267 proteins, and they have improved from the initial GOR 1. And for GOR 4, they have an overall 64% accuracy. mind it, they are predicting turn as well. So they are predicting alpha helix, beta sheet turn, and so the prediction accuracy is a bit on the lower side, but the methodology is also very interesting, and this is an incremental method. So people have started following this, and in the GOR 5, they have actually utilized a PSI blast, and they could further enhance the mean accuracy to around 73.5% and SOV score to 70.8%. So as you can see that GOR methods are still in use, and they are continuously working on these methods to improve it further. One of the very latest methods which is considered as one of the best so far is called PorterPaleAle. So they are using bidirectional recurrent neural networks, and the accuracy they are achieving is in excess of 82.5%. It's a complex method which uses many different informations, many different physicochemical properties, and again it is based on PSSM based features. So PSSM based features, and they are tuning free parameters in the order of 13,000 to 18,000. So 5 architecturally different bidirectional neural networks are trained for each fold, and then cross validation, and they used an ensemble based approach to finally predict the residue information, the secondary structure information for each residue. And it is giving very high accuracy, and they actually have 2 different data sets. The data sets and 2 different servers as well. The data sets of Porter and PaleAle 4 are sorted by the set of quality. See, in bioinformatics and in all the data sets as we are discussing here, they have many different quality. As you can see that somewhere I have mentioned that the high resolution data. So how accurately we can actually have a secondary structure information of a experimentally validated structure is an important stuff. So what is the quality and what is the resolution? So some people use very high resolution, for example less than 1.8 angstrom. So that means with less than 2 angstrom difference, even in less than 2 angstrom difference also, you can classify, or you can identify the shape of the structure that correspond to a particular residue. And in some cases, if the resolution is not so good, then you can say, okay, I can do it at 5 angstrom resolution or even more. That means there may be slight errors in the final annotation. So they have actually classified the overall data into 2 quality, that is high res and low res data, and the combined. So they have reduce it to 25% sequence. We work with that, and sequence similarity threshold they have used 25%, resulting in 9,152 proteins, and further selected 7,522 proteins with a better quality, and remaining 1,630 proteins of lower quality for testing purpose. So they have finally used 7,522 proteins and used 5-fold cross validation on Porter on high res, giving 81%. As you can see that the full set training, if you use the full set for training and only high resolution set for testing, the secondary structure prediction accuracy is around 82.3%. So you can see that if you predict on you train on the full set and predict on the low resolution set, the accuracy is very low. So if you train only on the high resolution set, then you can see that accuracy is actually a bit lower side, because you are not getting the full training information. So in general, if you train on the full set and also test on the full set, you are still getting 82% accuracy, which is really commendable achievement. And this is, as I said, this is one of the best methods available till date. PaleAle is a completely different server which actually predict residue solvent accessibility. So this is a separate work which is we are not discussing here, but again this is the I'm just discussing that this is predicting the solvent accessibility of each residue, and this is also a server. They are using the same data set to predict this solvent accessibility of each residue. But as you can see, the accuracy is a bit on the lower side, but that is not an important point to be discussed here. As I said, that Porter server is very popular, and even you have a lot of direct bindings available using Python and all, so that you can actually use these Porter server predictions for any unknown sequence prediction task. So overall, we have discussed different types of secondary structure prediction methods, and as you can remember that in the first lecture it was a more generic discussion, and we discussed what do you mean by secondary structure, and what is alpha helix, what is beta sheet, and how we can actually have very different very basic secondary structure prediction methods, like say starting with Chow-Fussman's method to hidden Markov model, how we can actually model HMM state diagram and have some basic idea of secondary structure prediction method in a classical sense. But here today we are actually we have discussed that how we can use these secondary structure prediction methods coming forward from the classical approaches, and what is used today, because secondary structure prediction method is a very classical problem is with the ever increasing sequence to structure gap. Like there is always a huge requirement for efficient secondary structure prediction methods. So how we can, like a method called PSIPRED, how it actually take a simple sequence information and extract some standard features like PSSM and some physicochemical properties, apply some standard machine learning algorithm, apply windowing technique, apply a two-stage cascading strategy or a consensus strategy, use high resolution set, low resolution set, and finally maybe design a web server so that someone can submit there and submit to your server and get the prediction done. So all these things are covered, and I think this gives an overall idea about how this particular domain works. Thank you very much for watching.